Start, madam.
Okay. Good morning, students. This is Sarala, working as a JL Botany, Hasanparthy, Warangal. Welcome to Telangana Residential Educational Institution Society webinar online classes. It's given in this slide. They are Abhineshri, Harika, Archana from Vangara, Divyanjali, Mahalakshmi, Lakshmi from Chautupal, Nagashri, Akila, Triveni, Sadaf from Vaira, Bhavana, Sureka, Alochana, Surisha and Anusha from Balanagar. Okay, students, in previous class, we had already discussed about transcription and translation process. In those process, how proteins are synthesized from DNA molecule. Okay, today we have to discuss about regulation of gene expression. You already know about gene. Gene is the functional unit of the individual or functional unit of the organism. So regulation of gene expression means regulation of gene expression or gene regulation. It includes a wide range of mechanisms that are used by cells to increase or decrease the production of a specific gene products, nothing but proteins or some other substances. Gene regulation means it is a mechanism. This mechanism used by cells to increase or decrease the production of a specific gene product. Each gene having a specific function, and each gene consists of two alleles, one dominant allele as well as one recessive allele, and each gene is perform a particular function. So with the help of this regulation mechanism, these genes they can synthesize some specific products in increased manner or decreased manner. Okay, so in eukaryotes, the regulation could be exerted at 4 levels. Eukaryotes, you already know about eukaryotes. Eukaryotes means U means advanced, karyotes means nucleus, advanced type of nucleus. Nothing but nucleus is bounded by nuclear membrane. Those organisms are called as eukaryotic organisms. In this eukaryotes, the regulation could be exerted at 4 levels. The first level is transcriptional level. Transcriptional level means formation of the primary transcript. In eukaryotes, in a transcription process is completed, hnRNA is formed. hnRNA, heterogeneous nuclear RNA, is formed due to the transcription process. This hnRNA is immature RNA. So this hnRNA undergoes some post-transcriptional changes, and it can forms a matured mRNA. So this at this transcriptional level, formation of primary transcript, nothing but hnRNA formation is takes place. And the second step is processing level. Processing level means regulation of splicing. Splicing means hnRNA, it consists, our primary transcript or hnRNA, it consists introns as well as exons. Introns are non-coding sequences, they are removed, and exons are coding sequences, they are spliced together. They can form a mRNA molecule. That events are takes place at this processing level. At this processing level, regulation of splicing. Splicing means introns are removed and exons are arranged in a specific order. That process is called as splicing. That process is takes place at the second level. So in the third step, transport of mRNA from the nucleus to the cytoplasm. The total transcription process takes place inside the nucleus because DNA molecule is present inside the nucleus. From that DNA molecule, this mRNA is synthesized inside the nucleus. Once mRNA synthesis is completed, this mRNA is transported from the nucleus to the cytoplasm. Once it is transported into the cytoplasm, it is involved in the translation process. Is it clear, all of you? In eukaryotes, regulation process is completed or exerted through 4 steps. That is transcriptional level. Transcriptional level means synthesis of mRNA from DNA. And this at this level, primary transcript or hnRNA synthesis takes place, and it undergo processing level, and regulation of splicing takes place, and that splicing is followed by capping and tailing. Okay, and at the third step, transport of mRNA from the nucleus to the cytoplasm takes place, and the last step is translation. Translation is this step plays an important role for the protein synthesis. This translation takes place in the cytoplasm region. Okay, these 4 events are observed in the eukaryotic organism, but in the case of prokaryotes, there is no well developed nucleus. Only at the place of nucleus, only one DNA molecule, single stranded DNA molecule is present. So that's why transcription as well as translation both are takes place in the same compartment. And in some cases, transcription is not completed at that time. Translation is begins in some cases. So that's why in the case of eukaryotes, in the case of prokaryotes, these 4 events are not observed. In the case of eukaryotes, only these 4 events are there. Okay. Next, the genes in a cell are expressed to perform a particular function or set of functions. Genes, they are present in the cell. Okay. So it is the cell. So inside the cell, and cell wall is followed by plasma membrane, and plasma membrane is followed by cytoplasm. In the cytoplasm, different kind of cell organelles are present, like ribosomes, mitochondrion, and etc. etc. The middle portion is occupied by nucleus. Inside the nucleus, genetic material DNA is present on chromosomes, and on the chromosomes, genes are present. The genes in a cell are expressed to perform a particular function or set of functions. The genes in a cell are expressed to perform a particular function or set of functions. Chromosomes are the carriers, they carry the genetic information, DNA, and on that chromosomes, genes are present in each and every cell. They can perform the particular function or set of functions. For example, an enzyme called beta galactosidase is synthesized by E. coli. bacteria, E. coli, Escherichia coli. So in that bacteria, an enzyme that is beta galactosidase is synthesized by E. coli cell. So that beta galactosidase enzyme, it is nothing but beta galactosidase enzyme. It is used to catalyze the hydrolysis of a disaccharide that is nothing but lactose into glucose and galactose. Beta galactosidase enzyme, which is present in the E. coli bacterium. That beta galactosidase enzyme break down the lactose into galactose and glucose. Bacteria use them as a source of energy. Is it clear? Bacteria, nothing but E. coli bacteria, it uses this glucose as well as galactose as energy source for their metabolic activities. Hence, if the bacteria do not have the lactose around them, means surrounding environment, in the surrounding environment of the bacteria, it does not have the lactose, so it is used for and do not have the lactose around them to be utilized for energy source. They would no longer require the synthesis of the enzyme beta galactosidase. Listen carefully. This bacteria, it synthesizes the enzyme beta galactosidase, and this beta galactosidase utilize this beta galactosidase enzyme. It hydrolyzes the lactose into glucose and galactose. This bacteria utilize this glucose and galactose for the energy source. They would no longer require the synthesis of the enzyme beta galactosidase. So it is the DNA segment. So in this DNA segment, genes are present. So genes, they are the functional unit of the cell or organism. They are located on the DNA molecule. DNA is present on the chromosomes. So on the DNA, genes are present. This DNA is involved in the transcription process to synthesize the RNA. It is nothing but primary transcript. This RNA undergo processing control and it can form a matured mRNA. So this is DNA molecule. This DNA molecule undergoes transcription process to synthesize the mRNA, that is primary transcript or hnRNA. It undergo post-transcriptional changes, nothing but RNA processing control to form a well-developed or matured mRNA. And this matured mRNA, which is formed inside the nucleus, from that nucleus it is transferred into the cytosol, nothing but cytoplasm. And this RNA transport control means this RNA molecule is transported from nucleus to cytosol or cytoplasm. This RNA, mRNA, is again involved in the translation process to synthesize the protein. This total process is considered as gene regulation or regulation of gene expression to synthesize a particular product. It is either a protein or some other substance. Is it clear? Once again, I explain it. It is the DNA segment. And the DNA, the gene which is responsible for protein synthesis is present on the DNA. And this DNA molecule undergo transcription to synthesize the or to produce the primary transcript. It is incompletely developed mRNA molecule. It undergo processing to form a mature mRNA, that is nothing but a messenger RNA. This messenger RNA is formed inside the nucleus, which is transported into the cytosol or cytoplasm, and which is involved in the translation process to synthesize the protein. This total process is considered as gene regulation or regulation of gene expression. So, so this regulation of gene expression is clearly explained with the help of lac operon, nothing but lactose operon. So before this, the operon concept was proposed by two scientists. This operon concept was proposed by two scientists. One of the scientists is Francis Jacob. He was a genetist and a biochemist monad. Monad, he was a biochemist, and this Jacob is a genetist. These two persons, they can develop the operon concept. Operon means that it is nothing but a DNA segment. Operon, an operon is a functioning An operon is a functioning unit of DNA containing a cluster of genes under the control of a single promoter. Once again, I repeat the definition of promoter. Sorry, operon. An operon is the functioning unit of the DNA containing a cluster of genes under the control of a single promoter. It is called as operon. So, this total DNA segment is considered as the operon. Operon means it is the segment of DNA consists a single gene or a cluster of gene to perform a particular function for the formation of a particular product. Okay. It is called as operon. That operon concept was developed by these two persons, Jacob and Monod. So first, then we have to discuss about lac operon. So lac operon means lactose operon. This concept was developed by Jacob and Monod in E. coli bacterium. The lac operon consists of one regulator gene, the promoter gene, the promoter as well as the operator, and three structural genes Z, Y, and A. Okay, lac operon, it consists, this lac operon, it consists a promoter region, operator region, and structural genes. Students, observe here, these three are the structural genes Z, Y, A. This is the operator region and this is the promoter region. At promoter region, RNA polymerase enzyme is attached to the RNA molecule for transcription process. Okay. Lacoperon, it consists of one regulator gene and the promoter region and the operator region and three structural genes Z, Y, and A. Along with this, it consists I gene. I gene codes for the repressor of the lacoperon. I gene, it consists repressor. Repressor, it attached to the operator and it can prevents the total regulation of the gene. So this repressor is indicates with the help of I. Once this repressor is bind to the operator, total regulation of the gene expression is stop or ceases. Okay. So these are the parts or regions which are present on the lac operon. They are regulatory gene, promoter gene, operator region, structural genes ZYA and I gene code for the repressor of the lac operon. And there are three structural genes are there. They are ZYA. The first structural gene Z, the Z gene code for an enzyme beta galactosidase. This enzyme is responsible for the hydrolysis of lactose into glucose and galactose. Observe here, this is the structural gene Z, which is responsible for the synthesis of an enzyme beta galactosidase. The structural gene Z, which is responsible for the synthesis of beta galactosidase enzyme. This beta galactosidase enzyme causes the hydrolysis of lactose into galactose and glucose. And the next structural gene, and the gene Y codes for the enzyme permease, which increases the permeability of the cell to lactose. This gene Y, which is codes for the enzyme permease, which increases the permeability of the cell to lactose. Permeability means it can allows this permease enzyme, it can allows the cell into the lactose medium. So this is the structural gene Y. This gene is codes for the enzyme permease, and this enzyme permease, it can allows the cell into lactose medium. And the last structural gene A, and this structural gene A encodes an enzyme trans-acetylase. Okay, and the structural gene A, it encodes an enzyme. That enzyme is trans-acetylase. This is the structural gene A. So this structural gene A is encodes an enzyme trans-acetylase. Okay, these are the three structural genes which are present on the lac operon on the RNA molecule, mRNA molecule. They are structural gene Z, structural gene Y, structural gene A. Structural gene A is codes for beta galactosidase enzyme, which cause the hydrolysis of lactose into galactose and glucose. Structural gene Y codes for permease enzyme. This permease enzyme, it can gives permeability to the cell into the lactose medium. And the last one is structural gene A, which is codes for an enzyme trans-acetylase. Okay. Hence, all these gene products in the lac operon are required for lactose metabolism. These three structural genes, and promoter as well as operator and a regulatory gene, these are the main for the gene products in the lac operon are required for lactose metabolism. Lactose is the substrate for the enzyme beta galactosidase, and it regulates the switching on and switching off the operon. Lactose, it is the disaccharide which is acts as the substrate for the enzyme beta galactose or beta galactosidase. It regulates the switching on and switching off of the operon. So that's why this lactose is termed as inducer. It is means lactose. So lactose, it can plays a vital role in the switching on and switching off of the lac operon. So that's why this lactose is termed as a inducer, and this lac operon is called as inducible operon. Is it clear? This lactose is functions as inducer. It can induce the switching on and switching off of the lac operon system. So that's why lactose is considered as the inducer, and this lac operon is called as the inducible operon. Okay. Next, in the absence of a preferred carbon source such as glucose, if lactose is provided in the growth medium of the bacteria, nothing but E. coli bacteria. So, in the absence of preferred carbon source, such as glucose, in the absence of the glucose, then we have to supply the lactose to the culture medium of the E. coli bacteria. Then that lactose is transferred into the cells through the action of the permease. This permease enzyme is coded by structural gene Y. Okay, structural gene Y codes this permease enzyme. It can gives permeability to the cells to grow in the lactose medium. Then the lactose induces the operon in the following manner. Glucose is absent. In the absence of glucose, lactose is provided to the bacteria, and that lactose is transported into the cells through the action of permease enzyme, and that lactose induces the operon in the following manner. So, in the presence of inducer, means in the presence of lactose, this lac operon is switching on. Lactose is acts as inducer to switch on the lac operon. So, this lac operon, it consists operator region, promoter region, and regulatory region, and I region. And these are the structural genes which are present on the lac operon. So, operator region, promoter region, and regulatory region as well as structural genes are located on the lac operon. So, in this, lactose is available to the E. coli bacterium, and this lactose, it acts as inducer. This lactose is bind to the with the help of I gene. I gene is codes for the repressor. Repressor means it stop or prevent the regulatory mechanism. Is it clear? This repressor, it can repress the activity of the structural genes, and it can prevent the regulation of gene expression. But in the presence of this inducer lactose, this inducer lactose is binds to the repressor. This repressor become inactive. It does not affect the operon. So Then this RNA polymerase enzyme is bind to the promoter region, and the total transcription process is proceeds, and that lactose is breakdown into glucose and galactose. So in the presence of this inducer lactose is functions as inducer. It bind to the repressor, so that's why repressor become inactive. Then this lactose is entered into the cells with the help of permease enzyme, and that lactose in the presence of this beta galactosidase or beta galactose enzyme, that lactose is breakdown into galactose and glucose, and this transcription process is proceeds and proteins are synthesized. Okay. Next, if lactose is not available to the E. coli cells, lactose is not available to the E. coli cells. So, in the absence of inducer, means in the absence of lactose, this repressor it is active because there is no inducer. So in this case, inducer is there. This inducer is bind to the repressor. This repressor become inactive. But in this case, this inducer is absent. So that's why this repressor become active. This repressor is bind to the operator region, and RNA polymerase does not bind to the promoter region, and total transcription is blocked. Students, a little bit confusion is raised here, so that's why all of you listen carefully. So in the absence of lactose, this operon is switch off. So that's why this repressor it become active because inducer is absent. Inducer nothing but lactose. Lactose is not supplied to the E. coli bacterium. So that's why this repressor is active, which is bind to the operator region, and this RNA polymerase enzyme does not bind to the promoter region. So that's why the total transcription process is blocked. So it is about lack operon. So in the presence of lactose, nothing but it is functions as inducer, and the total transcription process is proceeds, and these structural genes are active and they can send codes for three particular specific enzymes, and lactose is breakdown into glucose and galactose, and it is the energy source for E. coli bacterium, and transcription is immediately followed by translation and particular proteins are synthesized. In this case, operon is switching on by the inducer lactose. So in this case, lactose is not available to the E. coli cells. So in the absence of inducer, nothing but in the absence of lactose, this lac operon is switched off, and the structural genes are non-functional, and the transcription process is blocked, and this repressor it become active and it bind to the operator, and it can blocks the total gene regulation process. The repressor of the operon is synthesized all the time from the isine. So this repressor is synthesized from the I gene, which is located on the operon. It is the I gene which is located on the operon. This I gene is used for the synthesis of repressor. Repressor of the operon is synthesized all the time from I gene. The repressor protein binds to the operator region of the operon and prevents the RNA polymerase from the transcribing the operon. In the absence of lactose, it is bind to the operator region and prevents the activity of RNA polymerase to prevent the transcription of the operon. But in the presence of the inducer, such as lactose, lactose is used as inducer and it is bind to the repressor and repressor become inactivated. RNA polymerase is activated or access the promoter and transcription is proceeds. Okay, so in this case, lactose is bind to the repressor, so that's why repressor become inactive and the transcription process is proceeds and structural genes become active and operon is switched on. But in this case, there is no inducer, means lactose is not available to the bacterial cell. So this repressor become active, it bind to the operator and it can prevent the activity of RNA polymerase and the total operon is switched off in the absence of lactose. So that's why lactose is very, very essential for E. coli bacterial cell and it functions as inducer. So that's why lac operon is called as inducible operon. Lactose is functions as inducer. Okay, so in the presence of inducer, nothing but lactose, repressor is inactivated and RNA polymerase to access the promoter and transcription process is proceeds. Okay, it is about total lac operon and regulation of gene expression is totally completed and then total molecular biology unit is successfully completed. Then coming to our third assignment. Okay. The first question is define the term operon. Define the term operon. Operon, nothing but the segment of DNA which consists cluster of genes. Those are responsible for the synthesis of a particular substance. It is called as operon. This operon concept was developed by two scientists, Jacob and Monod. And next, coming to the second question, which type of structural genes present in the lac operon? I already explained now. There are three types of structural genes are present in the lac operon. They are Z, Y, A. Okay. It is about second question. Next, coming to the third question. Explain the functions of promoter and operator. Promoter, it is the starting point of the operon and RNA polymerase is bind to the promoter and operator is also present on the operon. It can regulate the total transcription process. It is about third question. Next, coming to what is mean by charging of tRNA. This charging of tRNA, this topic is explained in the previous class, but that question is given in this today's assignment. Okay, it is about today's assignment. Okay, and thank you everyone. The total molecular biology unit is successfully completed. Thank you all.